Our next speaker is Britt Adamson. She's a postdoc from Jonathan Weissman's lab at UCSF, and she will be talking about using a multiplexed single-cell CRISPR screening platform known as Perturb-seq to dissect the unfolded protein response.
Okay, thanks for the introduction, and thanks to the organizers for inviting me to share my work today. I'll be talking, as was mentioned in the introduction, about our recent efforts to build a platform for performing single-cell gene expression profiling of pooled CRISPR-based screens. But before I get into the details of the experiment, I want to tell you a little bit about the biological question that motivated us to build this technology in the first place. So this question centers around the unfolded protein response, which is a branch stress response network responsible for buffering and maintaining the function of the endoplasmic reticulum. For the purposes of this talk, there are really only two things you need to know about the UPR. One, that although it's called the unfolded protein response, which is because it's been studied predominantly in the context of its activation in response to unfolded protein stress, it can respond to a diversity of inputs, some of which I've listed here. And from the ones I've listed, I hope you can appreciate that these can represent very somewhat diverse physiological conditions. The second thing you need to know about the UPR is that one of the results of activating the UPR is the induction of an integrated and dynamic transcriptional response. Now, this response is controlled by three transmembrane sensor proteins. These are ATF6, PERK, and IRE1, and they sit on the membrane of the endoplasmic reticulum. And upon sensing conditions of ER stress, these activate discrete downstream signaling events that initiate three corresponding transcription factors to translocate into the nucleus and induce gene expression profiles that are both separable but overlapping and integrated. So once you know these two things about the UPR, that it can respond to a diversity of inputs and that the transcriptional output is pretty dynamic, one of the big questions that becomes apparent is what is the logic behind the larger organizational structure of the unfolded protein response? And from an experimental point of view, we can ask the question, to learn about UPR organization, can we map the transcriptional outputs for each type of input stress? Now, to approach this sort of systems level question, we thought that this was this question would be really good to approach with genome scale genetic screens. I was really lucky because as I was thinking about this question was during a time when the CRISPR genome scale perturbation libraries were really coming to maturity. And we've heard a lot described about different types of CRISPR screens at this workshop. And so I undertook a traditional pooled genetic screen. This is a flow cytometry-based screen. And using the CRISPR interference perturbation libraries that were really beautifully validated and developed by members of Jonathan Weissman's lab. As I mentioned, it's a flow cytometry screen. And to perform the screen, I built a reporter cell line that expressed an M-cherry reporter gene under the control of an IRE1 responsive promoter. So when the cells encountered a genetic perturbation that caused ER stress that would induce the IRE1 branch, the cells turned red. And by looking at the population of cells that turned red, we could get a good idea of what types of genetic perturbations cause this type of stress. So traditional pooled genetic screens are a really fabulous way to identify sets of hits genes, and this screen was no exception. You can see in the results of the screen where each black dot and peach dot represents genes targeted by our library. We identified a set of about 400 genes that had increased reporter signal with above a threshold level of significance. But when we looked at the data from this screen, one of the things that becomes very apparent when you do this type of screen struck us, which is that what we measured here were really low complexity phenotypes with no power for determining the relationship between the hits in our gene set. And this is perhaps best illustrated by thinking about an example. I keep hitting two buttons instead of one. And so what I've illustrated here are three of our top scoring genes, which are the three core subunits of the translocon Very briefly, the translocon is a protein pore that is in the ER membrane, upon which the ribosome docks, and through which nascent proteins are inserted into the ER membrane or the ER lumen. And what you can see is the three components, Sex61 alpha, beta, and gamma, scored very strongly in the screen. But the only reason we knew that these genes that were hits in our screen were connected to each other was because previous biochemical studies and a lot of previous work had identified that these were subunits in one protein complex. But what we didn't know is how these translocon genes relate to the rest of the hits in the library. And not only that, because the phenotype we measured in the screen is just the ability of disruption of these subunit genes to induce a M-cherry reporter, which is supposed to stand as a proxy for a larger transcriptional response. The other information we didn't have is what are the full transcriptional consequences of translocon depletion in these cells. And so what we thought, we thought about this for a while and realized that what we really wanted to build was a scalable screening platform capable of collecting transcriptome-level phenotypes. After building this platform, really think about, can we scale this to a point where you can think about doing high-throughput pooled screens that give you very complex phenotypes as your readout. So together with Tom Norman, who's another postdoc in Jonathan's lab, we built the Perturb-seq platform. And as I mentioned, here we're using CRISPR interference to give us our genetic perturbations. But especially since I'm at the Broad, I want to mention that Perturb-seq was built in collaboration with Aviv Regov's lab. And members of her lab published a companion manuscript in December with ours that used CRISPR cutting libraries to do this type of work. And that presents a very different set of bioinformatic challenges using those libraries. As I said, this work was published in December, and we tried very hard to outline all the technical details of how this platform works in those papers. So what I'll present here really is just an overview of what the technology looks like. We're using lentivirus to deliver pools of guide RNAs to our cells. Here, again, we're using CRISPR-I. And we're screening in K562s, which is a cancer cell line. And these cells carry the dCas9 dCas9 crab fusion protein necessary for CRISPR-I. So very briefly, by targeting the dCas9 fusion protein to promoters of target genes using the guide RNAs, we can inactivate transcription, and that's how we get our genetic perturbations. Now, in this platform, what we do is we take the perturbed cells and we put them through droplet-based single-cell RNA sequencing protocols. Now, for those of you who are unfamiliar with single-cell RNA sequencing, the droplet-based protocols basically sequester individual cells into oil-based droplets. And within these droplets, the cells are lysed and release the mRNAs. And then we can actually use these droplets as very, very tiny reaction chambers to do reverse transcription reactions. And in these reverse transcription reactions, the cDNAs are labeled with cell-specific barcodes. So when we get our sequencing back, what we actually get back are the mRNA identities that are linked to these cell barcodes. So computationally, we can stitch back together the transcriptomes that we've isolated from individual cells. Now, what Tom and I layered on top of this, of course, is the ability to identify the CRISPR perturbation that was delivered to the original cell. So now we've built a platform where we can get transcriptome-level information for individual CRISPR-based perturbations. So what we did, of course, because at the time I was studying the UPR, we did an experiment related to our traditional screen. And we took about 100 hits from our IRE1 activation screen and put them through the Perturb-seq platform. In this experiment, we analyzed about 65,000 cells, and we got a very, very high mapping rate for the guide RNA identity in each of the transcriptomes we got back. Now, because we were analyzing so many cells with a relatively small library, we had many replicate cells for each perturbation that we put in the library. And the first thing we did is we used this information to reconstruct synthetic bulk RNA sequencing profiles, and then asked a very logically straightforward question, which is, how do these synthetic RNA-seq profiles compare to each other? And so we built a correlation map of how all of the bulk RNA sequencing data sets relate to each other. And then by clustering this information, what I hope you can see is that we were able to identify groupings of genes or genetic perturbations that we analyzed on the Perturb-seq platform that were related through their biological function, either because they did similar things in cells, or as an example in the bottom where you can see the triple T complex, where they're actually part of larger protein complexes. And so this gives us a way to map how hits from a genetic screen are really related to each other and get a better idea of what the biological function of some of our hits are. But of course, we have transcriptome level information here, so we can also think about asking questions about the phenotypes themselves. And because we were studying a branch transcriptional response, one of the questions we wanted to ask is, can we look at the ways that these hits are activating the UPR, the branches of the UPR, independent of each other? So many of these hits, as I mentioned, were isolated in a screen where we took a measurement for activation of the IRE1 branch of the UPR. But specifically, one of the things we wanted to do next was ask what's happening with the other two branches of the UPR when we have these genetic depletion events. So we turned back to our Perturb-seq platform and did a little bit of a different type of experiment. Here we're analyzing a much simpler library, but we're doing it with combination of genetic perturbations. So in this experiment, we're delivering 1, 2, or 3 targeted guide RNAs to each cell, in an effort to knock down the 3 sensors of the UPR individually or in combination. We then take those cells and expose them to a chemical stress that causes strong UPR induction of all 3 branches and submit these to the Perturb-seq platform. And what we were able to get back from this experiment is really a profile of what activation of each branch of the UPR looks like. This is a T-SNE plot of the gene expression data set that we got back. And for those of you who aren't used to looking at this type of data, each dot on this plot represents an individual cell. And what I hope you can appreciate is those cells, based only on the unbiased clustering of the gene expression data set, those cells are really clustering both based on their chemical treatment, but also based on the guide RNAs that were delivered to each cell. Now, the colors on top of this plot are pseudo-colored based on our separate identification of the guide RNAs that were delivered to each cell. So this data set really represents our abilities to independently isolate the gene expression profiles involved in UPR signaling, but also validates our method very nicely for how well we're identifying the guide RNAs delivered to each cell. Now, we were really excited about this data set because what it allowed us to do is to have a ground truth for what UPR activation looked like. And we could use this data set as a training set for machine learning algorithms to teach them to recognize activation of the 3 UPR branches. We could then use that analytic pipeline to have those algorithms, to apply that to the sublibrary profiles we generated in our first Perturb-seq screen and ask specifically how do each of these genetic perturbations activate the 3 branches of the UPR. So the scores for activation of each of the branches are listed in the graph below here. You can see activation of the IRE1 branch, ATF6, and PERK. And what I hope you can see is that the different genetic perturbation events are really activating the UPR in different ways. You can see sets of biologically related genes here that activate IRE1 and PERK, but not ATF6, and other sets of genes that have differential activation. So we wanted to look a little bit more carefully at what some of our top hits from the original pooled genetic screen were, how they were performing. And we saw something very interesting, which is that these translocon subunits were really specifically, preferentially activating the IRE1 branch. Here you can see beta, alpha, and gamma, and you can see strong induction of IRE1, but minimal or no induction of ATF6 or the PERK branches. Now, this information was particularly interesting to us because in our epistasis experiment, what we saw is that expression of the translocon encoding mRNAs, or induction of those mRNAs, was a feature that actually defined IRE1 branch activation. So for all of the cell populations that were exposed to the UPR-inducing agent, but only those that had active IRE1 upregulated translocon genes as part of their UPR response. And what this suggested to us is that there was an isolated feedback loop between translocon expression and IRE1 alpha signaling. In other words, that when you have depleted translocon levels, you get preferential activation of IRE1. These are proteins that are all on the ER membrane, so they do have local access to each other. And that preferential activation of IRE1 could then upregulate translocon genes. And this was biologically very interesting to us, and we've subsequently done some follow-up work looking at this model. But what I wanted to point out today, what's really neat about this model is that it came out of only primary sequencing data. So to get from doing larger scale screens to really a biological hypothesis, we didn't have to invest that much time in doing exploratory validation studies. And so this will be my wrap-up slide for Perturb-seq. But one of the things I really wanted to stress today, which we think is a major advantage of this platform, is that you can get comprehensive information from primary screens, which we think makes it a very powerful platform. Other advantages are that you can do parallel RNA-seq in uniform conditions. I've shown that we can do systematic decoupling of transcriptional circuits. What I didn't have time to talk about today is that we were able to identify behaviors that are driven by subpopulations. Remember, we have single cell information here, so we can really subdivide the cells in many different ways. And also, that the platform requires relatively few cells, and so we think this makes it a really nice platform for thinking about doing genetic screens in rare or hard to isolate cell populations. With that, I'd like to thank everyone who contributed to this work. It was a large effort that required help from a lot of people. My co-first author, Tom Norman, did a beautiful job working on the analytical pipelines that we used. Jonathan has been a terrific mentor, and as I mentioned, all this was done in communication and collaboration on the platform itself with Aviv Regev's lab, and their lab published a really nice companion paper, as I mentioned, using CRISPR cutting libraries to do very similar things.
I really like this method, the Perturb-seq method, but I was wondering, all the data you showed us from 100 perturbations could maybe also be done in an arrayed format with single RNA-seq experiments. So I think the real strength of this method would be to scale up to like 20,000 perturbations. Does this work?
So there are some technical limitations to scaling with the quality of data that we're getting now. But we are pioneering methods now that we think are going to be really amenable for helping to really scale the platform. What I will say also is that there are two advantages to doing it, this type of work with even fewer guides in a pooled format over an arrayed format. One, since we were studying a stress response, these type of studies are often really affected by batch effects. And so by doing, even if you're just pulling out the RNA sequencing data, by doing it in an internally controlled environment, we got much nicer data. The other thing is that a lot of the analysis analyzing the individual activation of the branches did rely on the single cell information that we had. We needed, these methods really required having replicates of the individual gene expression data sets. Great, thank you.